So good morning, good afternoon, and good evening, everyone, depending on where you are joining to us today. I'm Neriman Yilmaz, and together with Dr. Marcus Birken, who generally hides behind the computer, we are hosting the TEBI International Seminar Series. So today, our speaker, before I introduce her, I just would like to tell you why we actually invited her, because on Twitter, on Sophien Kamoun's Twitter account, I saw a sentence and it was saying that plants lack an adaptive immune system. So Yorgos Korelis and Clémence Marchal in the Sophien Kamoun's lab had the ambitious idea to piggyback mammalian adaptive immunity and produce made-to-order plant immune receptor Pikobodies. When I read this, actually, I was mind blown. Like, who came with an idea like that? So basically, you are making a llama to produce nanobodies for plants so that plant immune can be developed. And so I was immediately found Clémence and then asked her, like, Clémence, would you like to give a presentation to us? Because I have millions of questions to you, but maybe if we make it to an international seminar series, then more people can ask the question. And Clémence was so kind to accept that. But she's not only kind, because she is also a very well-established person in this topic. So Dr. Clémence Marchal completed her PhD at the John Innes Center, Norwich in UK in 2019. And since 2021, she's a postdoctoral scientist at the Sainsbury Laboratory, Norwich, UK. She's interested in the molecular mechanisms underlying plant-microbe interactions and how to harness them to develop improved disease resistance in cultivated plants. So Clémence, once again, thank you so much for your willingness to give this seminar. And we are all looking forward to hear your very cool topic and how did you come with this idea?
Hi Neriman, hi everyone. First of all, thank you. Thank you so much for giving me the opportunity to share our work and talk about our story. And it's a real great pleasure to be here. Yeah, I hope you will be interested and I will be happy to answer any question at the end of the presentation. So today I'm going to talk to you about Pikobody, which are made to order plant immune receptors. So yeah, as Neriman mentioned, I'm currently in the lab of Sophien Kamoun at the Sainsbury Laboratory here, which is located in Norwich in the UK, at the east of the UK. And the main objective of my postdoc is to understand the molecular mechanism underlying plant pathogen interaction and plant immune receptor engineering. So before I start, I would like to stress that this is a team effort. So the whole lab contributed to this project, and particularly Sophien, of course, for hosting us and scientific contribution. But my co-first author, Yorgos, was actually the one who came up initially with the idea. Andres is a pre-doc in our lab currently, and he helped a lot with the experiments for the Pikobodies. And Adeline is our research assistant and contributed to many of the experiments that I will be talking about here. And also, this work wouldn't have been possible without the BLASTOFF group. So the BLASTOFF is a collaborative group between several labs internationally. So you have Mark, Nick, and Sophien here from the UK, Ryore Teroshi from Japan. And together, these groups really undergo like this research on the receptors, the wild-type receptors that I'm going to talk about today. And it's because of them and all the work that they've done that this engineering was made possible. Let's dive into the science now. So I'm sure most of you are aware that in the wild, plants are actually very efficient at fending off pathogens. However, in our cultivated plants, pathogen and pest remain a majorly constraint in agriculture. And that's what you can see on this heat map here, where the color shows the yield loss percentage based on cereal disease and in different regions of the world. So many diseases can can affect many crops, and it's a problem worldwide. So now there are several strategies to tackle this issue. I forgot. So the main issue is the way we cultivate crops. So using monoculture and developing resistance genes in a way that once we develop one single resistance gene in a field that applies such a strong pressure on the pathogen that it will heavily select for virulent races. And what we can see in the field is that the more we cultivate a certain cultivar that has a resistance gene X, the more we will start to see appearing in the field a pathogen that will become virulent to this particular resistance gene. So this is one of the main issues that we are trying to tackle here and to break the cycle. So there are different options that we have. We could obviously spray chemicals in the field. We could include more diversity using different varieties in the same field. But this obviously comes with drawbacks. So spraying chemicals is not really nice for the environment. It's really costly. Not all farmers can access to them. And just the same as for resistance genes that we can develop in plants, pests and pathogen can also develop resistance to the chemical that we spray. Regarding diversity, it's not really convenient for farmers to have different varieties because sometimes they don't have the same flowering time or the same seed setting time, so that can impact harvest. So what we could do is trying to develop, to deploy several resistance genes in the same crop or rotate between them. But identifying always new resistance genes can take a long time, even though we are getting better and better at this now. It takes a time to identify them, and it takes also some time to introgress them into our commercial varieties. So really our idea is how about designing them directly and hopefully being able to develop a system that we can quickly adapt to the pathogen populations that are present in the field. And the reason why we would need to design them is because plants don't have an adaptive immune system like us. So as if your immune system is challenged with a particular antigen, our cells, our immune cells will be activated and many division will be occurring with a somatic mutation happening in these cells, which will develop antibodies that have different sequences in their binding site to the antigen. But plants don't have that. One plant will be born with a set of immune receptors and will keep the same set along its life. But they are not helpless. So here is a cartoon showing a plant cell in green. So with the apoplast, the plasma membrane, and the cytosol here, and different pathogens are able to cause disease to the plant, principally via secreting small molecules, either in the apoplast or in the cytosol. And plants have evolved immune receptors to be able to sense this molecule and trigger an immune response. So we have some receptors that are located in the plasma membrane that are mostly involved in recognizing these molecules that are secreted in the apoplast. And we also have immune receptors that are in the cytosol, so intracellular, able to sense the pathogen molecules that are secreted inside the cytosol. And today I'm going to be talking about this class of immune receptor. And among these intracellular immune receptors, most of the ones that we characterized so far belong to the same family, which are NLRs. So NLR stands for nucleotide binding leucine-rich repeat immune receptors. And as you can see here on this graph that shows the number of resistance genes that have been cloned over the years, most of them belong to this NLR family. So NLR have a very conserved tripartite domain organization. So you have an N-terminal domain, either colloid or tier, that's involved in signaling and downstream response. The NB domain, that's really the hallmark of this protein family, is involved in modular activation of the receptor via ATP binding. And then the leucine-rich repeat domain in some NLR is involved in sensing the pathogen, so recognizing pathogen effectors, and also interacting with the other domains to maintain the NLR into an active state. So we are gaining more and more insight now as to how NLR function in plants and how they are activated. And we also like to categorize NLR based on how they work. So we have some NLRs that we call singleton because they can both recognize the molecule from the pathogen and trigger the immune response. We have NLRs where there is a division of tasks with one NLR that will sense a pathogen molecule, and this interaction between the NLR and the pathogen molecule will then go activate another NLR, and this other NLR will be in charge of triggering the immune response. So because this NLR is only involved in binding the effector, the pathogen molecule, we call it sensor, and the one that is required to trigger the immune response, is called helper. And now we have evidence, like phylogenetic evidence, that actually most of the NLR pairs that we see where there is a sensor and helper are derived, have evolved from a multifunctional ancestor. And we think that this division of labor allows for accumulating more diversity in the sensor, so it can keep evolving and recognize other pathogen molecule while still being able to interact with the helper, that this one which is able to induce the immune response. And that can lead to also very complicated and intricate networks. So in the lab, we work on a specific network that's called NRC network. So that stands for NLR required for cell death. And in this network, we have many different sensors that can recognize molecules from many different pathogens. And each of these sensors are able to activate one or several of the NRC helpers. And in return, these helpers can activate the immune response. So as you can see, we can have like very complicated interactions between these molecules. But today I'm gonna focus on a particular NLR pair when we have only one sensor and one helper. So I've told you that pathogen secretes effector in the plant cell to manipulate the host and cause disease. So they target specific host proteins to do this. And we have a specific class of NLRs that have evolved from integrating domains that are very similar to these host proteins that the effector trigger. And the idea is that by doing that, it will bait the pathogen effector and then use that to trigger immunity. And because these domains are non-canonical, so they do not belong to the typical domain that we see in NLRs, we call them integrated domains, and we call this NLR IDs. So NLR IDs are actually, we can actually see them very often in plants. They can make up to 10 to 5% of the whole set of NLRs of a given plant species. And the domains that are integrated are really, really varied. So we have very different domains that can be integrated. So as you can see on the word cloud here. And more strikingly, we also have allelic series of NLRs where the helper is really conserved. The sensor is also very conserved apart from this integrated domain here at the C-terminus. And these particular domains, so they are depicted in orange here on this cartoon, can be integrated at various places of the NLR. They can be at the N-terminus, they can be in the C-terminus, they can be in between signaling domains as well. So in the lab, so in Sophien's lab, we are mostly interested in several NLR pairs from rice that all have an integrated domain in the NLR scaffold. As you can see here, so HMA, the HMA domain here and the NOI domain here for PII. And all of these NLR recognize effector from Magnaporte oryzae, which is the rice blast, which causes the rice blast disease. And strikingly, all of these NLR recognize these effectors via their integrated domains. And then they require the helper to be able to trigger the immune response. So since NLRs can accommodate integrated domain in their scaffold, why not design our own? So why not take this domain here that's present in a wild type and engineer it to develop new resistant specificity to pathogen? And this concept, we didn't come up with it. Many people have done that in the past and still doing that as well. So targeting these particular integrated domains to expand the recognition spectrum of these particular immune receptors. But the question that we asked ourselves was, what would be the ultimate integrated domain? So something that would be short, so it doesn't disrupt the overall structure of the immune receptor, easily adaptable, so we would be able to switch between integrated domains very easily without causing major disruption, and also stable, so that it wouldn't prevent the protein from folding properly. And the one domain that came up to mind was nanobodies. So nanobodies are a specific type of antibodies that are found in camelids. So that's like your alpaca, llama, or camels have nanobodies. And basically, there are antibodies that only have a heavy chain, as you can see here, compared to the ones that we can find in humans, for example, that have heavy chain and also light chain. And the fragment that's responsible for antigen binding is actually very small. It's only 15 kilodalton. So just over 100 amino acids. So it fits the bill of like being short. And just so you can see a structure of a nanobody. So here in gold is the scaffold. So that's never changed from one nanobody to another. And these variable loops here are the regions that are responsible for antigen binding. So the region that will have most of the diversity across nanobodies. So this is how the Pikobody concept came about. So we were working on these rice immune receptors that have integrated domains that sense pathogen molecules. And we thought, can we actually swap these for a nanobody? And so there are many nanobodies that are from which the sequence is publicly available. And these are particularly binding the green fluorescent protein and cherry fluorescent protein, because they are used in microscopy a lot. For example, enhancer apparently enhances the fluorescence from GFP, which is really useful for microscopy approaches. But for us, we thought that it would be a very cool proof of concept if we could use these nanobodies and integrate them into the NLR scaffold from PCAM. So one of the rice immune receptors I was talking to you about, and see whether this would be functional, if this chimera would be functional. So when I talk about Pikobody, it's the functional unit of the sensor PCAM1 that has the nanobody integrated at the place of the HMA domain and the helper PCAM2. And so this work has been pre-printed first on bioRxiv. It was available on bioRxiv and is now recently published in Science. So you will see a lot of leaves in this talk. So I just want to make sure that everyone is familiar with the assays that we are conducting here. So we evaluate immune response in Nicotiana benthamiana via agroinfiltration. So that means that we transform our NLR and our effector of interest into different agrobacterium strains. And then with an infiltration buffer, we resuspend this agrobacterium, and with a syringe, we infiltrate a leaf. And then we look 5 days after infiltration whether we have cell death or not. So in the ideal case, infiltrating the NLR alone or the effector alone wouldn't cause any cell death. And when we put the two together, we have recognition of the effector by the NLR that triggers cell death. So in this case, cell death is a good thing because that means that we have an immune response. However, very often when we start swapping domains or making mutation in NLRs, we can make them becoming autoimmune or autoactive. So that means they will trigger cell death even in the absence of the effector. Right, so the first thing that we did, before even challenging the Pikobodies with the fluorescent proteins, was to see if these Pikobodies were autoimmune or autoactive. So in this particular leaf here, each infiltration spot shows a Pikobody with a particular nanobody integrated in the absence of GFP. And here PChem and AVR PICD, so that's our positive control. So PChem is the wild type with the HMA domain, and AVR PICD is the effector that it recognizes. So it's just to show that the assay works. So here we can see that both minimizer and LAG24 Pikobody were autoimmune, but we had 3 Pikobodies here that were not. And we also tested, as I mentioned, some Pikobodies targeting M. cherry. And again, we had some that were autoimmune already and some that were not. So the next step was to use the Pikobodies that were not autoimmune and to challenge them with the green fluorescent protein or M. cherry to see if they would trigger an immune response. So here, for example, an answer challenged with GFP triggered an immune response, but not with M. cherry, which was the ideal case. So that was the perfect scenario for us, because that means that the chimera is likely sensing GFP and able as well to produce an immune response. We had some that were non-functional at all, like this one. We had LAG16 that was functional. And the autoactive LAG24, well, actually, if you look at the cell death, it's actually a lot stronger in the GFP infiltrated side than in the M. cherry one. So it could still be functional, but it's still a bit autoimmune, so we don't really like that. And the same for the Pikobody M. cherry. We tested different Pikobodies against GFP or M. cherry. And in some cases, like LAM4 here, or LAM8 here, we had a specific response to the fluorescent protein against which the nanobody was raised. So that was really encouraging, because that means, okay, that's great, that works. But something that was bugging us was, can we actually reduce this autoactivity? Because if you can see here, like there is still a response to GFP, but can we get rid of the autoactivity here? And very luckily, there was a paper published while we were doing this work showing that we could improve the stability of nanobodies expressed in plants by targeting specific mutation in the scaffold. Oh, getting dark. In the scaffold of the nanobody. So here they pointed out some particular residues that were associated with a decreased stability of the nanobody in the cell. And when they mutated them from other residues, they could improve the stability. So we did the same in our Pikobodies. So as you can see here, we took LAM4 as an example. Our original LAM4 was autoactive because it triggered response both to M. cherry and GFP. But when we introgressed these mutations that were identified by this group into the scaffold of LAM3, we could completely abolish the autoactivity here to GFP while maintaining the response to M. cherry. So that was really great, and that showed that we can actually recover some autoactive Pikobodies from mutating the scaffold, which doesn't interfere with the variable loops here. Right, so, okay, it works in Nicotiana benthamiana while overexpressing GFP or M. cherry. So for the follow-up experiment, we focused our effort on one nanobody that targets GFP, so that's enhancer, and one that targets M. cherry, which is LAM4. So what was next is, can they actually trigger an immune response in the presence of a pathogen? The only issue is there are not many pathogens that secrete GFP or M. cherry. So we had to engineer a virus, here potato virus X, to make it produce free GFP or free M. cherry and use these to infect Nicotiana benthamiana. And then we used both Western blot to detect GFP and M. cherry, and also fluorescence, since both are fluorescent protein, as a proxy for viral load. And that looks like this. So in this leaf, we are looking at GFP fluorescence. So here, when we infiltrate PVX GFP with the empty vector, so that's nothing, is only the backbone, we have strong fluorescence. And when we co-infiltrate the virus with Rx, so Rx is a resistance gene that recognizes PVX, we have reduction of GFP fluorescence. When we infiltrate enhancer, we also have similar phenotype than for Rx. And when we infiltrate LAM4, we have fluorescence. So we have virus, which is expected since LAM4 doesn't recognize GFP, but recognize mCherry. And just additional controls, we had the wild type PChem, so the one that recognizes the effector from Magna Porte. So it's normal that we see accumulation of PVX. And here was the infiltration buffer only. So if we translate that into histogram of the intensity of the fluorescence, we can see that Pikobody enhancer reduces the amount of PVX in the leaf as much as Rx, whereas Pikobody LAM4, well, since it's a PVS mCherry, behave the same as the wild type. And just to check, we also did a Western blot to check for protein accumulation, so accumulation of GFP. And we can still see a bit of GFP here, but it's very reduced compared to the wild type or even Pikobody LAM4. So this was great because that means that potentially this could actually confer resistance against actual pathogens. And one of the other questions that we had is that can we stack Pikobodies? So that means, can we express different Pikobodies on the same vector without impairing their function? And the idea behind that is that it's harder for a pathogen to overcome several resistance genes. So one of the approaches is to express several resistance genes onto the same vector and co-deliver them all into one plant to potentially increase durability of the resistance. So can we actually do that with Pikobody as well? And the reason why we ask this question is that sometimes when we mix together different alleles of the same NLRs, we have a dramatic effect in the progeny. So for example, for the dangerous mix alleles, so if these two are combined in the F1, you can see that the F1 has a dramatic developmental defect compared to the parents. And we even have the case for so here it's for powdery mildew resistance genes. When specific alleles of powdery mildew are combined in the F1, for some reason, one of the alleles suppresses the effect of the other. So the F1 remains susceptible against particular virulent strains. So because of this, we wanted to test whether the Pikobodies were still functional when they were co-expressed. So first of all, are they compatible and do they have additive effects? So the first question was, are they compatible? So here we can see that yes, we can co-express Pikobody, co-infiltrate Pikobody in Nicotiana benthamiana, and this still doesn't cause HR or immune response. This is our positive control, so that's great. We won't have the case of the plant that has like a dramatic phenotypic effect likely in transgenics. And then do they remain functional when they are co-expressed? And the answer is yes. So when we co-express one Pikobody and the wild type version of Pik, we have a response to mCherry but not GFP, and the other way around when we co-express enhancer with the wild type. And then when we co-express both Pikobody together, we have a response to both GFP and mCherry. So that's great. So that means that one, they can work together and they both still trigger HR to their corresponding effector. And we also, of course, tested in the presence of PVX, so in the context of an actual infection. And then again, so against PVX GFP, every sample that has Pikobody enhancer expressed leads to a reduced viral load to the same level as the resistance gene Rx. But when an answer is absent, we have GFP fluorescence, which means that we have virus replicating in the leaves. So the next step was, so all of these were transient assays in Nicotiana benthamiana. So the next step for us was to see if this still holds true in stable transgenics. So here I'm showing different transgenic lines expressing the wild type PCAM and different independent transgenic lines expressing or not, you will see the Pikobodies. And so this is just to show you that there is absolutely no phenotypic effect, at least on the plant growth, derived from the overexpression of the Pikobodies. And then when we look at the expression of the receptors themselves, so the line here is for PKM1, so that's the one that has the nanobody, the sensor. So we could see that we could find it in these 3 lines, but not in line 4. So for the next slides, you will see that line 4, we use it as a negative control. We see the wild type PCAM. Rx was not tagged with flag, so we don't see it here. And the wild type obviously doesn't have the receptor. And then when we look for the helper that was tagged with HA, we can detect it in 9.1.10, but very, very, very low in 4. And again, we see it in our wild type PCAM, and we see Rx in the Rx transgenic line as well, but nothing in the wild type. So then we use these lines for assays. So we challenge them with PVX via wound inoculation. So the way this experiment is done is that we take up the agro that expresses PVX on a toothpick, and then we pierce little holes on the leaf. And 1/2 of the leaf was infected with PVX GFP, the other half with PVX M. cherry. And then we only wait 7 days and we sample around the inoculation site to detect whether or not we have a virus. And this is the results. So there was PVX GFP in all the samples, and here are the lines at the top. So we could detect the virus in the wild type, so untransformed Nicotiana benthamiana, and also the Nicotiana benthamiana line expressing the endogenous, the wild type PCAM. And in our line, Pikobody and answer 4, because if you can remember, that was the one that was not expressing PCAM1 and only lowly PCAM2. But all of the lines that expresses both receptors, we cannot detect any virus, just the same as for Rx. However, when we look at PVX M. cherry, so since these are Pikobody and answer line, and an answer targets GFP, as you can see here, we can see virus in all of the lines here, just only not in Rx. So it's functional in stable transgenic lines, and it still remains specific to the particular virus strain that expresses GFP. And all right, so with that, our idea now would be to identify pathogen effectors in pathogen populations and be able to purify these proteins and express them to immunize llama. And then from the llama's blood, we can isolate nanobodies that are binding these particular pathogen effectors. And then we would have a system, because we have the NLR scaffold already, we would have a system where we could easily swap nanobodies. So going from one nanobody to another inside the NLR to confer resistance against plant pathogen. And with that, I wanted to say again, thank you to my lab, and thank you to you for your attention. Oops. And if you have any question, please let me know.
Thank you so much, Clémence. And it was really like, so even more mind-blowing.
Oh, thank you.
Yeah, so I will open the floor for the questions, but I think before, since I'm moderating this, I will take the opportunity to ask the first question. So I want to ask you, what about this technology and how do you think they will consider it a GMO or not? So, you know, if we would like to use it in the future, what are your thoughts in which class it will be classified?
So it will definitely be considered GMO. Yeah, because we are bringing a completely exogenous domain into a plant protein. So I think it will be definitely classified as GMO.
Okay, thank you so much. And I see. And so another question of mine that, for example, can we do something like that for a seed-borne pathogen?
From a seed-borne pathogen?
Yeah.
So I guess it will all come down to whether these NLR immune receptors are expressed in seeds. That's actually something I do not know. Because we usually look into leaf, shoots, and roots.
Yeah.
I actually never looked into seeds myself, but I would assume that if we have the receptor expressed in some tissues of the seed itself, and depending on like what the pathogen is infecting, like what tissue the pathogen is infecting in the seeds, it could be possible. I don't know if we can, for example, use like specific promoters that are expressed in this leaf tissue and put this in front of the NLR to drive their expression onto these tissues as well. The only limiting factors for the Pikobody is that it has to be secreted inside the cell because NLRs are intracellular immune receptors. So for now, we haven't explored using the cell surface receptors, but that's something that we are thinking of doing as well. But so far for Pikobodies, as they are, the limitation is that the pathogen molecule that they sense needs to be secreted inside the cell.
Okay. And we have a question from David, and he's asking that which chromatic type did you use in this study and why?
Okay, so in this particular, so we used already published nanobodies. I don't know if that was clear, like from the talk. So they were all derived from llama, if I'm not mistaken, because I think LAM like stand for llama, and LAG is also for llama. So in this case, we use these ones in particular because they were already published. They were shown to be expressed in plant cells because they are used a lot in cellular microscopy. So that's why we use them, because we knew that they would likely be expressed. But you don't have to use animal immunization to isolate nanobodies. Now they are actually synthetic nanobody libraries that are available on the market, where there is actually phages displaying the nanobodies that were synthesized, and then you bring your protein of interest and you screen for binding. So basically, I think it's a huge bank of nanobodies with a conserved scaffold and then some codon shuffling or like amino acid shuffling at the hypervariable loops to have almost all combinations you can have. So you could even bypass the animal immunization step by doing that.
And I have other questions. Okay. So I see that, Sander Mayburg from Heavy also, he is asking that, would you target conserved part of the pathogen molecule or more rapidly evolving sequences?
So I think it would be great to target core effectors, so effectors that are conserved among different isolates of a particular pathogens, just because likely it would be able to allow us to like have a resistance that targets different isolates. Now, Pikobodies won't solve the fact that obviously when you deploy them, you still apply a huge selection pressure on that particular pathogen population, and that will also select for virulent strain. The winning point of Pikobodies is that since we can easily swap between and choose which nanobody we want to integrate in the scaffold, we could easily adapt to whatever pathogen population is in the field. So we will need to couple this with pathogen surveillance in the field to be able to detect what effectors are present and then adapt the Pikobodies to it.
Oh, thank you. And then there's a question from Bernard, but maybe Bernard, you can ask it. since he is here.
Okay.
Our video is not going on. Yeah, Clémence, thank you for a fantastic talk. I'm wondering whether there is any reason to believe that the pathogens might evolve resistance either more slowly or faster to these than conventional resistance genes used in plant breeding.
So I think if Pikobodies are used as single resistance genes, they will probably the pathogen will evolve the same as for any like qualitative single resistance gene that would be deployed in the field. Now we could combine different Pikobodies on the same vector to be able to make them a bit more durable, but intrinsically it will be the same, exactly the same as for any other resistance genes. The idea here is that we can easily change the nanobody that's integrated. So hopefully we can keep up with the pathogen evolving via doing that.
Right, thank you.
Thank you.
Thank you. And we have a question from Maya Badri. Sorry for the pronunciation, but he said that thank you for the nice talk. Do you think this system could be used with singleton LRR by replacing the LRR domain with a nanobody?
That's a great question. It's really funny because NLRs are actually very finicky. Like when we start meddling with their domains, often we cause autoimmunity. And this is why we targeted this specific class of NLRs that already have other domains integrated in their scaffold, and that doesn't seem to bother them in their function. We haven't tried replacing the LRR with a nanobody. I would imagine that if we replace the whole LRR, we might interfere with the regulation of the LRR in the resting state, the NLR in the resting state, because we know that LRR domains sometimes interact with other domains to keep the NLR in an inactive form, and then that's binding of the effector disrupts this, and that's what activates the NLR. So I'm really just speculating here, but I would imagine if you remove the whole like LRR domain, we might have something that's always active, maybe, and not necessarily only like active when the antigen is here. But if you want to try, please do and let us know what you get.
Sort of, I say collaboration there. So, and Clémence, you showed that it worked for the virus. So, did you ever try any fungal species as well?
So we haven't tried that yet, but we did receive some nanobodies targeting Diomycete Phytophthora infestans. So we are currently testing this in the lab. So we received them not so long ago, so it's not ready yet. But then we would be able to test if they really confer resistance against a pathogen. So because the limiting factor also is that you need to be able to purify the effector, to purify the protein, and that's this that you send to the company, or you can also screen it yourself in your lab if you can buy the library. But you still need the purified protein to be able to identify nanobodies that bind to it. Okay.
Actually, you make it look very easy. So, but from starting to ending product, how long more or less it takes for example to develop if you have the receptors and
well, so now that the receptors, like now that the scaffold is well defined, I think the major part of the groundwork is done. Identifying nanobodies, I think it really depends on the company that you use and whether they use animal immunization or screen against a synthetic library. Because obviously, if they screen against a pre-existing synthetic library, then it's faster than having to immunize the animal and waiting several weeks that the animal develops like the nanobody against it, and then you have to isolate the sequences from the blood. But for example, like the company that we used, I think they took 2 months to send us sequences of nanobodies that were targeting our effector. So probably from one company to another, it takes more or less time. And then there's the matter of, well, testing them. So cloning them into the scaffold and testing that quickly in Nicotiana benthamiana. But this can be done very rapidly now.
Yeah. And the floor is still open for other people to ask questions, so don't feel like I am the only one. But if there's no other question, I have one last question as well.
Of course.
So that you show that you can, for example, for one organism, you can introduce multiple receptors and basically target those different parts. But can you also do, for example, one of the diseases that I wrote, sorry, you wrote, is caused by 2 different fungus. So can I then design for 2 different fungus as well, like 2 different fungi as well? Or like, can I combine them, two of them together, or do I need to design 1 for 1, 1 for 1?
Oh, you mean having one nanobody that binds to 2 effectors?
Yes.
That's a really good, we haven't tried this. I would assume that it's probably a bit more difficult to find such nanobody that can simultaneously bind the 2 proteins. So I think it would be more likely that you would have to have 1 nanobody for one nanobody for another, and then put the 2 Pikobody on the same cassette, on the same like backbone, to be able to co-express and co-deliver them.
Okay. Okay. Yeah. Actually, you answered all my questions. Thank you so much.
Right.
But are there any other questions? The floor is open. But if not, as you see, Clémence is quite communicative person through Twitter as well. You can ask her any questions that you would like to ask. I believe she is one of the easy to reach people, definitely. So thank you, Clémence, again. And I am now handing it over to Bernard to make the closing.
Okay, thank you very much.
Clémence, thank you again for a fantastic talk. I was just starting to type another question in here, a more general one, Clémence, to ask, what was the original trigger for the idea?
Sorry, can you repeat? I didn't get the last part.
What was the trigger for the original idea? Because
the trigger, how did we think about that? Okay. So it was actually during an informal joint lab meeting between different labs from like Sophien allies network. That they were thinking about, that would be cool if we could have put an adaptive immune system in plants. Like, can we provide the plant with antibodies? Because then it would be really easy to adapt this to whatever pathogen we need to target at that time. And I think so it stemmed from this particular discussion among lab members. And then I think the idea of nanobodies was also thrown out at that time, but obviously we didn't really know how to make that. And that's Jorgos that then used these receptors, these rice receptors, because here on site and also other groups all over the world use these receptors to do domain swapping to try to expand the resistance to other pathogens. So we thought, maybe I could swap it with a nanobody. I don't know. I guess we won't know it until we'll try. And it did work.
Yeah, absolutely fantastic. And thank you for sharing that with us and the work. It's creative, it's inspiring, and obviously very powerful. We look forward to seeing how this will further develop in the next coming years. I think illustrating the growing power of synthetic biology for protecting our plants as well. And really fabulous to have had this directly from you this afternoon. Thank you for spending the time with us and for everybody that's joined us. And Merriman and Marcus have organized it. It's been stimulating and really much appreciated. All the best for your future work. We look forward to following this as it develops.
Thank you so much. Thank you for having me, and thanks everyone for joining and for the really nice discussion afterwards. Really cool.
Thank you again, everyone, and hope to see you next time. Bye-bye.
Bye-bye.
Bye.
Bye-bye.